Hi everyone, this is topic 12C, lymphatic system. This is the third and final lecture on lymphatic system. We have already started learning about adaptive immunity, which is the specific defense, and this is the third line of defense. If we classify adaptive immunity based on the type of cells involved, then there are two types of adaptive immunity: humoral and cellular immunity. Humoral immunity is also known as antibody-mediated immunity because this is produced by antibodies which are present in blood and body fluids. And in this kind of immunity, the B lymphocytes are involved. B lymphocytes, the B stands for bone marrow derived lymphocytes. Cellular immunity is known as cell-mediated immunity because here antibodies are not involved. The kind of lymphocytes that elicit this kind of immunity is the T cells or the T lymphocytes. T stands for thymus-dependent lymphocytes. That means these lymphocytes have to go to thymus gland, get the hormones, thymosin and thymopoietin, to mature into adult T cells. This figure shows how both cell-mediated and antibody-mediated immunity act together when antigens enter or attack your body. The cell-mediated immunity will mount a direct physical and chemical attack on the antigen, while the antibody-mediated immunity will activate B cells. The B cells will produce antibodies. The antibodies start circulating in your bloodstream, and then the antibodies go and attack the antigens. This slide shows the development of the two types of lymphocytes, B and T lymphocytes. You know that all blood cells start their journey inside the bones. So the bones have the stem cells for development of blood cells. From the stem cells, pre-B cell and pre-T cell, which are immature B and T cells, they are developed. Now the pre-B cell can mature into an adult B cell inside the bone marrow. And the mature B cell is known as an immunocompetent B cell. Then the mature B cell will go into blood circulation and go to different parts of the body, including lymph nodes. On the other hand, the T cells cannot become immunocompetent inside the bone marrow. So they go out as a pre-T cell into the blood circulation. Blood circulation takes it to the thymus gland. Inside the thymus gland, in the presence of thymosin and thymopoietin, pre-T cell matures into T cell, and then the T cell again goes out via blood circulation. It reaches the lymph node. So lymphocytes, once they have become adults, they are known as immunocompetent. However, they are still regarded as naive immunocompetent because they haven't encountered the antigens that they're going to fight with yet. So the naive immunocompetent lymphocytes, they reach the secondary lymphoid tissue such as lymph nodes where the encounter with antigen occurs, and it's only when the lymphocytes have encountered the antigens and they are binding with their recognized antigen, that's when they become fully functional antigen activated immunocompetent cells. These are the real soldiers who have the skill to fight against invaders. This figure shows the journey of the T cells and the B cells starting from the stem cells in the red bone marrow all the way up to when they become fully functional antigen activated immunocompetent cells. Now we are going to learn about humoral or antibody mediated immune response. When an attack occurs, the B lymphocytes, there are different types of B lymphocytes and they all have their specific receptors. So depending on the type of invader, a particular B site with specific receptors would bind to this specific antigen. And this binding causes or starts a clonal selection process. Here, a large number of clones would be produced. That means the kind of B cell that can fight that specific invader, they would be produced in large numbers. This is known as primary humoral response. Most of these B cells clones would become plasma cells, and plasma cells have the ability to produce antibodies, and antibodies are those proteins which circulate in your blood and they can destroy the antigens. Some of those B cells would not become plasma cells, they would become memory cells, and the memory cells are responsible for starting a secondary humoral response anytime an encounter occurs in future. This graph shows the primary response and the secondary response. The secondary response is faster, longer lasting, and stronger than the primary response. This figure illustrates the antibody mediated response. Antibodies are known as immunoglobulins, or in short we write it as capital I small g small s Igs. So these are soluble proteins which are secreted by the plasma cells, which are a kind of B cells, and the antibodies circulate in blood plasma. They act by binding to specific antigens. Anatomy of antibodies or structures of antibodies. Antibodies are made up of two pairs of protein chains. That means there are four polypeptide chains, and these chains are connected to each other by special bonds called disulfide bonds. One of the pair of the chains is longer, it's called the heavy chain, while the other pair is shorter, it's called light chain. Each chain contains two regions, a variable region and a constant region. Variable region is the one that varies in structure. This is the part that combines with the antigen, and because there are so many different types of antigens, so variable regions have to be different from each other because they have to bind to different types of antigens. The constant regions, on the other hand, are similar. They are same in antibodies, and they are mainly responsible for maintenance. They are responsible for activities of antibodies like complement activation or attachment. This figure illustrates the Y-shaped structure of an antibody. There are 5 classes of antibodies or immunoglobulins: IgM, IgA, IgD, IgG, and IgE. Remember the mnemonic MADGE, M-A-D-G-E. Each of these classes have different roles. Antibodies have 7 functions, or these are the 7 steps by which antibodies inactivate antigens. First is it binds to the antigen binding sites and neutralizes it. Second, it causes precipitation and agglutination, that means clumping of the antigen and helps in formation of immune complex. Third is it activates complement proteins, which if you remember are a group of 20 proteins. Fourth, it attracts various types of phagocytes such as macrophages, microphages. Fifth, opsonization, that means it flags. So once it has bound to the antigen, it calls for backup help. Sixth is it stimulates inflammation, so there is an inflammatory response. Seventh, it prevents further bacterial and virus particles to come and stick to these antigens. This figure illustrates the 7 functions of antibodies. Moving on to cellular or cell-mediated immunity, which is brought about by T cells. Here, for the T cells to act, they must be presented with the antigens by other cells such as macrophages. So a macrophage would present an antigen to the immunocompetent helper T cell. This is known as antigen presentation. The helper T cells are like scouts. They are the first one to know where trouble is, and then they are the ones who signal other T cells such as the killer T cells, also known as cytotoxic T cells, to come and start attacking the antigen. The helper T cells not only signal to the killer T cells, but they also call the B cells, and the B cells would come and they would change into plasma cells and they would make the antibodies. This figure illustrates antigen presentation. There are 3 types of T lymphocytes. The first one is the killer or the cytotoxic T cells, which are the soldiers who engage in direct face-to-face combat. So they specialize in killing infected cells. They make a certain chemical called perforin. The second category is helper T cells. These are the scouts. These are the recruiters. They go and they see, the first ones to go and check where trouble is, and they are the ones who recruit other T and B cells. The third type is the suppressor and the regulatory T cells. These are the cells that would release chemicals to stop the fighting of T cells and B cells. So they suppress the activity. And in each of the class, few members would become memory T cells. This figure shows how a killer T cell or a cytotoxic T cell would attack an infected cell. It has a two-pronged approach. First, the cytotoxic T cell would secrete a chemical called perforin. Perforin makes holes on the cell membrane of the infected cell. So the ECF fluid rushes inside the infected cell and it lyses the cell, so the cell bursts apart. Secondly, the cytotoxic T cell would also produce an enzyme, and this enzyme promotes apoptosis. Apoptosis means cell suicide. So this enzyme would go inside the cell and would repeatedly tell the infected cell to commit suicide. And so it's kind of like attacking from the outside, and also inwardly it is counseling the infected T cell to kill itself. This is an integrated summary of the immune response. Another figure showing integrated immune response. We are done with immunity, and now we are going to learn about organ transplants. And you must have heard that rejection is very common in organ transplants. So first, the major types of grafts or transplants. First one is called autografts, where a tissue is transplanted from one part of the body on another part of the body of the same person. Okay, so the risk of rejection is minimal. The second one is called an isograft, where identical twins are taken and tissues or parts of the body are transplanted from one twin to another. Now, since identical twins are genetically identical, this also has minimal rejection risk. The third one is an allograft, which is the most common one, where tissue or an organ is taken from an unrelated person. And the last one is called a xenograft, where a tissue is taken from a totally different animal species. So that means a human might get a structure or an organ not from a human, but from, let's say, a baboon or from some other animal. This figure shows the different types of grafts. Allergies are also known as hypersensitivity. These are disorders of our immune system. Here it is an abnormal vigorous immune response to antigens because haptens have complexed with our own proteins. There are three types of allergies. The first one is called immediate hypersensitivity, which is very fast, and this is caused by the release of a chemical called histamine, and this happens because IgE antibody binds to mast cells. So most of the anti-allergies that you see over the counter available in pharmacies, these are antihistamines. The second type of allergy is called a delayed hypersensitivity, and this is caused by the release of another set of chemicals called lymphokines from activated helper T cells. And in this allergy, the symptoms usually appear a few days after contact with an antigen. The third type is the dangerous one. This can be fatal. This is called anaphylactic shock or anaphylaxis. This is a systemic response. The entire body responds against the particular antigen. This figure illustrates the onset of allergies. Another disorder of immunity is immunodeficiency, where your immune cells or complement production becomes abnormal. This could be genetic or this could be acquired. An important example is AIDS. AIDS stands for acquired immunodeficiency syndrome. This is caused by a virus, HIV, human immunodeficiency virus. Autoimmune diseases are also disorders of immunity where the immune system cannot distinguish between self and non-self antigens, so it starts producing antibodies and attacking its own tissues. The table here provides a list of some common autoimmune disorders. For example, if you see hemolytic anemia, this symptom would be fatigue, weakness. Here the autoantibodies would destroy own red blood cells. Or rheumatic fever, where the autoantibodies would attack the heart valve cells. Ulcerative colitis, where the autoantibodies would be against the colon cells. Effects of aging on the immune system. Your thymus starts decreasing in size, so there is less space available for T cells, less hormone available for T cells to become mature, so you are losing some of your most specialized soldiers. Immune surveillance decreases. Primary and secondary responses, they take longer time, so now more antigen is required to produce the same response. Antibodies are produced in less number, fewer memory cells are produced, so in general your immune system is getting weaker.